Everyone, the meeting is being recorded.
Yeah, JB, you're ready to go.
Cool. Thank you. Again, as my usual introduction go, we're going to we have Erin Dickie here. My introductions are always very kind of a protocol, I would say, not following a protocol. What I want to say about Erin is that first, she's obviously a very academic, excellent professor at the CAMH. That's for sure. She's done much more than that, and that's the thing that I want to emphasize is that everyone has a very stellar academic record and all those things, and that's for sure Erin has. But she's worked so hard on the open science aspect and the teaching aspect that is really complementary. It's like the whole spectrum of activity that have been extremely impactful. So I'm going just to cite some of the open science who've been the brain hacker person for a long time now, and first probably the one starting to organize it in Toronto. Again, at the open science SIG involvement in the standardization of our data aspects and open open standards, and then on the teaching side, incredible. I think that's kind of big as well. But all the work that your lab and yourself have done on the software carpentry. I don't know if you guys have heard about the software carpentry, but it's a wonderful organization and exceptionally good teaching material that are open to everyone and all those things. So Erin is all of that, and of course other things in the teaching aspect, like I think you've been at academy as well, and maybe
Oh, actually, no, I haven't done
yeah,
that.
okay, well, anyways, you should have.
Yeah.
But plenty of initiative that Erin has done. So I'd like to thank you very much. There's a lot of things that are good to hear from you, and also again on this kind of a broader aspect of science that Erin has embraced. Cool. Thanks very much.
Yeah, thank you, JB, for the amazing introduction. I'm going to come back to some of the excitement of open science at the end of the talk, but I decided I was going to start with my bread and butter kind of research scientifically. And I realized that you guys, the QLS seminar has a very broad audience, so I thought I need to start with just generally an introduction to two basic concepts, or two concepts that I list in this title, which are neuroimaging and mental health. So I'll start with mental health, because I think I'm the only QLS speaker who's talking about mental health
This year?
this
Possibly.
year, actually,
Yeah.
or at least in the list so far. So I thought,
I think Satra had a little bit of a mental
Oh, okay.
but
Yeah.
not much. So you should be good.
okay, so I want to start with a little bit of thinking about what is mental health, because this is actually where I sit thinking. This is where now I'm actually thinking maybe too much about what is mental health. But let's start with about, so one way to think about mental health, and maybe that's the wrong way, but a way that tends to get tagged is mental health as the absence of mental illness. But then we have to think, what is mental illness? So mental illness currently and probably for a while later is defined based on clinicians giving people diagnoses, and they do this based on diagnostic manuals. So the current manual is DSM, Diagnostic and Statistical Manual 5. Diagnostic manuals use clusters of symptoms. They ask people about symptoms, and then they decide if they have enough symptoms from category A, B, and C, then they can get this particular diagnosis. Okay, so if you try to diagnose all the symptoms, so there's one study recently that actually repeated this in the community 4 times over 4 decades. So for every person, they asked about all the symptoms, they diagnosed all the potential problems. So I'm going to ask the group, guess how many people had at least one diagnosis in their lifetime. That's over 4 decades. Percentage. So how many of the people tested? This is from the community. If you ask people about mental health symptoms 4 times in their lives, all the symptoms, all the potential diagnoses, or most of the potential diagnoses, how many do you think had a diagnosis at least one point?
Was it both of them?
It's close. It's 80%, actually. So it got pretty close to all of them. And this is like, yeah, a clinician level classification of a diagnosis. This is only asking 4 times in people's lives at specific times for research. From that study, additionally, they saw that most of the diagnoses that occurred, at least the first diagnosis that occurred for that person occurred usually in adolescence. But that's not the diagnosis that they had for the rest of their lives. Diagnoses could change over time. Also having multiple diagnoses was actually more common than having one diagnosis. And having one diagnosis can mean a lot. I just want to stress that having one diagnosis can mean a lot of different things. So 2 different people with the same diagnosis could have a very different clinical profile. I'm going to get a lot more into that later. But also the diagnoses could change over the lifespan is an important thing to think about. Which is focusing on this now, I think the diagnoses are really important. And they still are the way that mental health, it's the way that clinicians need, it's what clinicians need to ask to figure out what treatments to prescribe. So they're still really important. But they don't necessarily show us the whole picture of what's happening at the individual level. Okay, backtracking. So that's my really fast forward interest to mental health, mental illness. I'm also going to give a super fast interest to MRI because I know that that QLS can have a lot of different students from a lot of different areas. So today in my talk, I'm going to be talking about 2 types of MRI or magnetic resonance imaging of the brain. The first is anatomical MRI. That's the one that gives you the picture on the slide. It's the one that you think about when you like Google MRI images of the brain. This is an image I got by Googling MRI images of the brain. But it's really useful because you can run automated pipelines and you can figure out making a lot of different measurements about the size and shape of different brain structures using this. The other one, which is my favorite, or used to be it's still my favorite. It's something called functional MRI. And by functional, use functional in quotation marks, we're really looking at blood flow across time in the brain. So as the brain gets used, blood flow changes. We can, back in the day when we started, we can compare blood flow at 2 different points in time, like related to a task, and make blobs on the brain to say which parts of the brain were involved in different tasks. The other thing we can do, which is much more my favorite thing to do, I don't know if it's came out nicely, is that you can actually just take the time series of the blood flow over time and you can correlate it with the blood flow from other regions of the brain. That generates a huge correlation matrix. The one on the left, this is the correlation matrix you get essentially for the parcellation. A correlation matrix on the right is essentially for the parcellation on the left, but you can get even more and more dense. So when people started actually looking at patterns of correlation across the whole brain, this is about 15 years ago now, we started really understanding kind of really cool principles about how the cortex potentially is organized by just looking at how different parts of the brain, which parts of the brain tended to actually have blood flow patterns that synchronize with each other. And this was about 15 years ago, 2011, we had like the Yo7 network or 17 network definition of the brain, where we started seeing parts of the brain that were far apart that we consider potentially parts of the same brain network. More recently, but actually, I guess, oh gosh, 8 years ago now, we also said we can take that matrix and we can decompose it in different ways. So when we did the networks, we were decomposing the matrix into subnetworks, subregions. But we said actually we can do different ways to decompose that, to use the connectivity matrix pattern to try to understand the structure of the brain. And this one uses a function called diffusion map embedding to try to understand other different link dimensions. We call them gradients of the brain. And these gradients were really cool because they actually made us think about where these networks are placed and how they might relate to each other. And it also gave us the ability to potentially, for every single point on the brain, put it in like a new space, essentially based on its relationships to others. So this is looking at this matrix decomposition. Okay, I will come back to this view. Okay, so now we have really cool, I just introduced mental health as a really important challenge. And I've introduced brain imaging a super cool tool, that's been around for like 30 years. So I guess my question to you is like, where do you start from there? Where do you come up with your next research questions? Anybody? Yeah,
Sure, I mean, how do different patterns of, brain activation correlate with clinician level symptoms or diagnoses?
yeah. One place to go. So, yeah, so one thing I wanted to stress, and I'll come back to it, is it surprised me that if you want, when you have these things, you want to think the coolest thing for you to figure out what to do, which may be like feed a cool deconvolutional neural network based on the neuroimaging to find diagnosis, might not necessarily be the most useful thing for a clinician, because a clinician can already figure out diagnosis. So it's very useful actually now. I feel like more of my time is spent talking to clinicians about what the research questions should be than it is about actually doing really cool analysis, which I apologize for, which means I feel like I have less cool analyses to show on my slides, but it was just sound. But I want to talk a little bit about the questions that we end up asking and the question and why we ask them. So the first part of my talk, I'm going to talk a little bit about parsing heterogeneity within disorders, because I already told you that two people can actually have completely different symptoms and actually have the same disorder. This for diagnostic classification, there's a lot of heterogeneity even within the disorder pool, and we don't currently really understand whether or not they really are subtypes or whether or not they are different treatable aspects of the disorder. The other thing I really want to talk about, this concept of looking upstream. So by the time clinicians end up seeing people, it will not be too late for them, but they might have been seeing mental health services for a while, because one disorder tends to turn into another. So some of the people working in some of the more severe disorder clinics, like psychosis, those people might have actually been seeing mental health services for a while, and we might have been missing them the whole time. So now the idea is to look more longitudinally. And the other one we'll come back to JB and mine's favorite topic, which is when you use the tools of reproducible science to put all these things together. Okay, so I'll start with parsing heterogeneity within disorders. Okay, so this is a great quote because it's true. If you met one person with autism, you've met one person with autism. The idea is that within especially some of these diagnoses, there's a lot of different people out there with a lot of different situations going on. Okay, so actually I'm going to talk a little bit about one disorder, not autism, but I'm going to talk about schizophrenia spectrum disorders, which affect quite a few of the population, but actually a smaller, maybe only about 1% or 1.5% of the population at the time, but are major sources of burden and need. And I'm specifically going to talk about cognitive deficits within the disorder or cognition within the disorder, because cognitive deficits are a core feature of schizophrenia spectrum disorders, but they're actually something that's still kind of untreated by the medications. So medications can make hallucinations and delusions go away for a lot of people, which is really helpful, but there's a lot of variability in the population about how much they're actually struggling to function in society. And social cognition kind of has to play a huge role there. So with this in mind, I think it was 2015, a while ago, right before I started at CAMH, 3 centers had banded together with funding from the NIH under the RDoC initiative. So RDoC is for looking for dimensions within disorders, to really focus on understanding social cognition within people with SSDs. So they worked together with a 3-site NIH grant. They each recruited 100 people with schizophrenia spectrum disorders for 300 people total, and 200 healthy controls. They were all actually stable at the time, but they did a decent kind of assessment of a decent, relatively long assessment for social cognition, long assessments for neurocognition, so non-social cognition, as well as clinical scales. There was also an imaging component that included social cognitive tasks. I think that was the main thing. And yeah, it was actually the first time that we really looked with all these social cognitive tasks together within a large population, a large 3-site study of people with schizophrenia. So when we had this data, one of the first things that we saw, or one of the first things that we looked at, was the relationship between these cognitive variables to each other. And one of the first things that we saw was that social cognition was actually really important, that actually impairments in social cognition were potentially more important to everyday functioning than the neurocognition. They tended to mediate that relationship in a path analysis. Although those things, social cognition and neurocognition were really correlated. The other thing we wanted to look at right away was this idea of looking at differences between people and parsing heterogeneity between people. And the way that we did that was kind of a series of clustering, data-driven clustering papers using the neuroimaging data. And we found these cool data-driven clusters. Viviano published on one right before he moved to Montreal and started working at Milla, or started training at Milla. And Colin Hocko, who I still work with this day, published a second clustering paper. These clustering papers were really interesting too, because the data-driven clusters we found that were important when it came to cognition and were related to the brain activity weren't actually specific to schizophrenia. They bled into the health control group as well. But recently, data-driven clustering is useful, but because it's not actually not diagnosis, but also because the clusters were kind of not that separated from each other, we started thinking about other ways to decompose the matrix or other ways to live more within the population. So more recently, I got a new postdoc. Seems like a new postdoc, but she's been around for a while. Ruchi Yu, who is actually was actually an expert in herself in partial least squares. So we decided to try to look at brain and behavior using partial least squares correlation. So what she put so many animations, and I'll just I'm sorry, I'm gonna go through. Okay. So partial least squares correlation, as everyone heard of partial least squares before. I'm going to give a super basic intro that it's multivariate technique. So you have a table on the left, a table on the right, and it's maximizing covariance. It's finding latent dimensions that maximize covariance between the two tips or between the two latent dimensions. You can find any number of latent dimensions. The analysis I'm going to talk about only found one, but it could find multiple. And then once you have those latent dimensions, you can get scores for individual participants. So brain scores and behavior scores by taking the loadings in those latent dimensions. And you can also go back and look at the loadings of the latent dimensions in the original input data. So she ran this analysis using that SPINS data set we talked about. On the left, so the behavioral variables that she brought into the X table were all the tests that were done for social cognition and neurocognition. So there were 15 or 10 or 15 different tests there. And on Y, we brought in those gradients of functional connectivity that I talked about a few slides ago. So we brought in the first three gradients. So for every region of the brain, we had kind of 3 scores relating to where they were in functional connectivity, where that region of the brain is in functional connectivity space. Okay. And what came out in this analysis was one major latent dimension, which generally associated pretty much all of cognition, both social and non-social cognition, with generally the brain makeup in a way that if you actually trace from case to control, or from those with the worst cognition to the ones with the best cognition, that's all these arrows. So these arrows kind of represent the loadings of each region in the brain, going from one along the latent dimension with the length of the arrow, strength of the arrow, being whether or not the loading was significant. And what you kind of just see across all these brains, all these regions, is all the regions all the arrows are pointing towards the middle, meaning that kind of the entire functional connectivity matrix is getting less differentiated or less followed apart. And these arrows, especially that are pointing towards the middle, over here and here, it's especially in that second gradient dimension, and that's especially the differentiation between the sensory parts of the brain. So between your motor and your auditory cortex and your visual cortex. It's actually really interesting to think about. What was also interesting is that when we took this out and started looking at it at left out variables, we looked at just functioning in the real world. This is from the Birchwood functioning scale and the quality of life scale. We saw that this poor cognition variable is associated with poor functioning, but also the brain variable that's associated with it in QLS. We also saw it being associated with negative symptoms, but not positive symptoms. Which is really interesting because sometimes when you just think about schizophrenia, all you think about are the positive symptoms, but this dimension, sorry, do people know what I mean when I say positive versus negative symptoms? So yeah, it's not about the hallucinations or the delusions, it's about everything else that you're stuck with. Why is this important? Okay. I'll take a break. Okay. And why is it important to look at social and neurocognition coming back and understand the brain? Well, what's really cool is that I still work with clinicians who are really trying to innovate in this space. So pretty early when we had some circuits that we knew were involved in social cognition, the clinicians came back and said, is it possible to actually target them and try to modulate them using transcranial magnetic stimulation? So this work is still underway. We still started clinical trials where we're actually within the same team, within the same network of clinicians, and we're now running a clinical trial of neuromodulation of the social cognitive circuitry in people with schizophrenia spectrum disorders with the hope that we can actually improve their social cognition. I was telling Nikhil that this is amazing, but also a pain in the butt because it's the first time that we need to do it. Because it's the first time or now the second time that we're actually need to have our pipelines and our computations work within a week, within 1 week turnaround so that we can get the scan, run the scan, test the scan, get our coordinates and give the information back to clinicians so they can start the treatment within a very fast timeline. And we have some really cool pipelines going on here where we're using both the functional connectivity patterns, so individuals' functional connectivity patterns of the areas of social cognition, but we're also, a really important part of the TMS is actually the folding patterns of the brain and just the structure of your skull can actually have a huge impact on how much stimulation you can actually get through the skull and to the brain. And actually what angle, because the brain is folded underneath, it ends up hitting the gyri has an impact on a stimulation current. So we're modeling that in combination with the functional connectivity patterns to try to get an individualized target. And we're doing that with 1 week turnaround within the process of a clinical trial. So we just finished a pilot trial and we're going to start the next one. And we're just starting kind of a larger, fuller base trial that's sponsored by Wellcome Trust. Okay, that was parsing heterogeneity within disorders, specifically looking at cognition within people with schizophrenia. Next, I'm going to talk about looking at screening. Okay. So I told you this, but we were actually surprised. There was a while ago, this is all coming from, this is clinicians' idea, and I was really excited to be there. But we knew that psychotic disorders were important, but we also knew that people with psychotic disorders, actually, the first time that they talk to a clinician about mental health is not when they experience psychosis. It's actually earlier on. About 75% of people who end up getting treatment for psychosis actually ended up getting mental health treatment for something else within the 5 years prior to the psychosis diagnosis. So this made us really think like that people who are struggling were hiding in plain sight. Because the people with the other diagnoses were actually just sitting in the other building. They were already coming to our clinics, and we had this area where if we knew how to intervene, if we knew how to predict what was happening, we might be able to make a huge change. So this is pretty exciting. I feel like we got the whole hospital together. We got like a solid like quarter to a third of all the scientists in the hospital participating in this large study. It has over 40 scientists, I think, on the investigator team. It also has an army of paid study RAs who are doing the assessments. And importantly, we also have the hospital's youth and family engagement core who are really helping us drive this study using impact from people with lived experience. Oh yeah, and it also brings together all the clinics with a lot of people with different types of scientific PhD only kind of neuroinformatics scientific expertise. So what is this exciting study? So this study is called the Toronto Adolescent and Youth or CAMH cohort study. The idea is that we're going to, we have funding from the CAMH Discovery Fund. the idea is that we're going to recruit 1,500 youth between the ages of 11 and 24. They're all seeking treatment at CAMH, but they're seeking treatment for multiple different things. And we're going to follow them for 5 years. We're also assessing people at multiple different levels. So we do that full diagnostic interview, actually pretty regularly, I think every 2 years. We also ask a whole lot of different questionnaires so that we have dimensional ideas about their mental health all the time. We're also asking about social determinant factors. We're also collecting, in Ontario, you can collect the OHIP number and you can link that to actual data before the study and after the study. We're assessing cognition, but most exciting for me, we also have this bioassessment that happens annually, so 6 times during the length of the study. What's in this bioassessment? Well, exciting for me, there's an hour of MRI imaging. The MRI imaging, for those of you who don't know, MRI imaging has sequences that are somewhat matched to the ABCD project, with the idea that we can kind of incorporate ABCD data as we go through. We also offer EEG for people who don't want to do the MRI, who can't do the MRI yet.
Sorry, just abbreviation version, what is RSA?
RSA is respiratory sinus arrhythmia. So it's essentially like heart rate monitoring, trying to get vagal tone and parasympathetic activity. Or I think it only takes like 5 minutes, 5 minutes of heart rate improvement. We also collect urine for a drug scan, or to try to understand medications better than participants might understand it themselves. And we also collect blood or saliva for people who are uncomfortable or refuse to give blood. Okay. So last year, ending last fall, we did a large study. So we're currently at around, I think, 700 recruited so far. The aim is to keep going to 1,500 and to hit that potentially in the next 2 or 3 years. We've been accelerating pretty quickly. And last year, we decided to do some pilot analyses to understand the characteristics and to publish the protocol for the first 417 people who got into the study. So this slide is essentially the demographics of the first 417. You can see we have a variety of ages, but we're especially hitting people who are just finishing high school, which will actually be interesting to see if we can keep them. What's also important is that we've been working a lot with the youth and the youth engagement core to really understand how to measure and how to report on gender identity. Because you know that within the world, this is actually kind of a moving construct. But we are noticing that if we try to meet students where, we try to meet our population where they're at, actually a large proportion, like 28%, are reporting that they come from a gender diverse background. Okay, in terms of diagnoses, it's all the things. This is from that diagnostic interview. I'm gonna pull again. So this is all the disorders present. With there could be any, so 80% have an anxiety disorder, 73% have a depressive disorder. So most of them have both anxiety and depression. How many disorders do you think each person has on average?
The paper said 3.5 on average.
Yeah. 3.5. Yes, you read the paper. Awesome. Okay. Yes, 3.5 on average. Also kind of critical to realize how much need there is that 44% actually endorsed clinical level suicide ideation within the last year. So they're really struggling at the point that they're coming to the study. The other thing that we tested for that we ask about, or that we're really interested in, is psychosis spectrum symptoms. So psychosis spectrum symptoms are not necessarily having a psychotic disorder diagnosis. It's endorsing any of these statements, essentially. So how many do you think endorsed at least one of these strongly, or 3 of these moderately or somewhat agree? So we have this classification of PSS, which is you have to endorse at least one of these strongly or 3 of these statements moderately.
What would you need to actually be given a psychotic diagnosis. Rather than
Worse
just
than that, yeah.
spectrum.
So, we can compare. So I'll give you the answer is that with the threshold of at least one of these symptoms or much moderately, it's about half the population.
Hmm.
Versus a psychotic diagnosis, we saw in about 8% of the population. So, that's quite a difference right there. And actually, we don't know whether or not these things are going to persist, whether or not they're just related to severity in general, or whether or not they really are markers for something worse at this point, because they do change over time. And actually, one of the points is to look at how they change over time. But one marker, but it is true that within the group who have this PSS classification, who endorse these symptoms, we see across the board everything else being related in pretty much the whole data set. So higher suicidal ideation, higher symptom severity on all of the other mental health scales, pretty much, and poor cognition. Okay. The other thing, which I'm pretty excited to announce, is that at the same time, I started doing a deep dive on the neuroimaging data and essentially just its quality, and how useful it would be. I think that's in one of the papers that I sent out. We're really excited that we had 80% of the people, so over 90% actually consented to participate in the imaging, so the MRI or the EEG, and about 80% completed. And when we deep dived, it was more about missed appointments and scheduling issues. That was why they didn't end up doing one of those assessments. We also got a decent spread of blood or saliva, but it's actually in the blood or saliva that you see kind of a switch where only about half of the people with the diagnosis are really interested in giving you blood. A lot of people feel sick issues there. Okay. And with the MRI data, there's some analyses that started recently. Has anybody heard of the term normative brain modeling? Yeah, in the back. Okay. So normative brain modeling or reference cohort modeling, as I saw one person call it, and I decided we should call it from now on because I just don't like the word normative. I don't like talking to youth with the word normative. Anyway, so reference cohort monitoring in general is you use fancy computational models or machine learning to actually, and a lot of data. So a lot of data across the lifespan to try to plot the aging curve across the lifespan or the development curve across the lifespan. So this is an example of a pretty classic development curve schematic that came from this paper by Bethlehem. Once you have this really cool aging curve model, you can take that model, you can apply it to new data. And what I'm really interested in is actually the error in the model fit. So if you train a model on exterior data and then you apply it to new data, within the new data, within the new samples, how much error is there in the model? So if this is the model and this is the new participant, there's two ways that we can really measure that error. We can do a centile score, which is really asking about where that person is in comparison to their age and sex matched peers. This is kind of cool because when we talk to other people about it, we were like, it's kind of like a height or weight curve that you have in the doctor's office, except now it's about your brain development. The other way that we've been looking at this is actually you just look at error in the Y direction. So if you project from the current model to where people are in age, and then you just subtract that projected age from their actual chronological age, you get something called the brain age gap, and that becomes another variable that you can start interrogating. When we interrogate this, when it relates to schizophrenia, there's now multiple replicated accounts of something called accelerated brain aging in schizophrenia, where the brain age gap, it seems to always be larger in people with schizophrenia. This seems to be across the lifespan, and actually, this is some data from our lab where when we looked actually at the brain age gap kind of curve across the entire time, it seems like the brain age gap or the accelerated aging might be happening before we even assess people, so before the symptoms start, because it seems to be there from a very early age. This is a more recent paper from Bethlehem, where he actually looked at centile scores, so the normative brain modeling across multiple disorders. And actually schizophrenia is the third most deviant or third most different disorder from the aging curve. And it's third most, the only other two disorders that seem to show more evidence for being off the aging curve were Alzheimer's disease and mild cognitive impairment, which are two that you kind of assume would be off the aging curve. Okay, so when we apply, so I'm going to start by the simple thing to do is actually now there's some really easy to use published calculators out there. You can just take the data from your neuroimaging models, you can plug it in, takes a summer student 1 week. It's pretty great because they just have to format the data and put it on a website and they get numbers back that they can do stats on. Two of the models out there are brain, and there's more out there now, but two that really work, especially in youth stats, are brainchart.io, which actually only uses 4 variables related to total brain. There's another called Napper, or age prediction R, and it uses 5,000 points across the cortex, but actually only the thickness of the cortex, and it calculates the age of the brain. When we applied this to the TAE data, across the board, looking at these kind of two ideas, centile score and brain age gap, I don't know how well you can see these, but we did actually see a significant difference based on that PSS classification. So that PSS being presence of psychotic symptoms. Presence of psychotic spectrum symptoms means when you have psychotic spectrum symptoms above the clinical threshold, you tend to have an older looking brain, even at that young age. Yeah.
Sorry, and your non-PSS cohort is still within your TAE
Yeah.
cohort.
Yeah. So they're still treatment seeking for mental health. Okay. So to recap the TAE study, the TAE cohort aims to better understand how certain mental health symptoms can develop and who's at increased risk over time. Early data from the TAE cohort study confirms that multiple diagnoses are present, and that nearly half are actually enforcing psychotic spectrum symptoms. And more recent data very early data looking at the MR data looks like psychotic spectrum symptoms like schizophrenia are associated with an older looking brain. When you're young, I don't know what it means to have an older looking brain, or whether or not that's pathological or whether or not that can change over time. But the fact that the TAE data is longitudinal is hopefully something that we can start looking. Okay, and one other shout out that's very exciting about the TAE cohort study is that kind of using the infrastructure and the ideas from the TAE cohort study, we've expanded. We just got funding from the Ontario Brain Institute to essentially expand something very similar to the protocol to 6 different recruiting sites, 5 within Ontario, 1 in Alberta, and a smaller proportion of those are going to be scanned. So it's very exciting. Youth mental health is on the outlook, and hopefully we'll have more answers soon. Okay, so that was my part about the excitement of looking at a screen. Now give me a moment to talk about JB and my favorite topic, which is reproducible science, reproducible and open science and tooling. It's good time. Okay, wait, I want to put this up. Who's seen Brenda Milner speak? Yes. I don't know, I was gonna say, does she still put up this picture? Yes, okay. I remember, so PS, I trained at McGill and back in IPN, and when I was in IPN, you get like 1 lecture from Brenda Milner. It was like my favorite thing ever. And I remember her putting up this picture. And do you remember what she does when she puts up this picture? She said that to me. Which was really like, I love that Brenda Milner, like with all of her amazingness, is there to give a shout out to all the people who inspired her and say how lucky she was to actually be training in the same room with Walter Penfield and Donald Hebb and involved in those studies. For me, I put up this picture. So inspired by Brenda Milner, I like to put up this picture. And does anybody know what this is? Yeah, brain hack Leipzig. It's actually the first brain hack. Everybody's in this picture. And it has these guys, who some of you might recognize. And it has all those people are in the picture somewhere. So these are all kind of, so this was near the end of my first postdoc. It was brain hack. And all these people, oh, there's Boris was there too. all these people really inspired me in terms of computational neuroscience, in terms of coding, but also especially in terms of reproducible science and open science, because some of the leaders of reproducible science and open science were all in this room. And then when I went to see them later, they're like, have you heard about this cool thing called BIDS? It's gonna change everything. We need to all do it. And everything went from there. Okay, and to speak to this, I wanted to say a little bit about the amazing neuroimaging data that already exists in the world. So I talked about some really awesome neuroimaging data that we're collecting at CAMH, but some of these other data sets are going to be really important. And I've done amazing work trying to make the data public and available. We have things like the adolescent ABCD, adolescent brain development, cognitive development work initiative, the Child Mind Institute and Child Mind Institute North, which is just some guys in an office in Montreal. Somewhere, I think. Yes, gotta find that. Okay, and Neuromod actually on the other side. Actually, Sam, were you involved in the first thing? Anyway.
Elizabeth.
Yeah. And yes, Neuromod, which is putting out an amazing amount of data from Montreal. That's great. Okay. But to make the most of the available data, and I feel like I'm preaching to the choir, you guys over there, you need to figure out how to actually get the data pre-processed in a standardized way or help people work with the data. And that's where standardization and reproducible science really come in. I got to give a shout out to Neurobagel, who's trying to push this forward, but also the original work, which came with the brain imaging data structure organization of the data, which was really important because before then it was the Wild Wild West. Everybody was calling one type of scan a different thing. And it was a pain and nobody knew how to run a pipeline the same way at all. And BIDS apps that really actually changed the thing. It said, if you organize the data, the app will know how to process it for you. So the app will actually read the data's metadata to figure out what the proper flags are to run, which was kind of a game changer in terms of teaching people how to analysis. I got involved in BIDS and JP. I got involved in this reproducible science world, mostly because I got up on a soapbox or got super excited about the idea that everybody who was doing functional or brain imaging analysis should be using the cortical surface more effectively. So understanding the folding structure of the cortical surface in their analysis. And there were a few different reasons for this. The reason why I think I got into it in the first place, although you can kind of fake it, is just prettier pictures, nicer figures. I'm going to say they're nicer, but they're also just more interpretable, which is really important too. When you can show brain activity just adds the entire cortex together, suddenly you don't need multiple slices, and suddenly a human brain can put together like whole brain patterns, which is really exciting. The second reason is that two-dimensional cortical registration. So everybody's brain is folded a little bit differently. So actually trying to register the brain using three-dimensional tools was kind of failing when it came to the cortical folding patterns. It was actually more effective to actually register the data on the surface, so to move along the cortical surface area. And the third, which is kind of a no-brainer, was that three-dimensional smoothing is actually bad. And so it boosts statistical signal. So any smoothing kernel will boost statistical signal. But when you smooth in 3 dimensions, especially in cortical areas, you're smoothing in white matter and you're smoothing in brain matter, and you're smoothing in gray matter that could be actually far apart because it's a little folded lump. Anyway, so you're smoothing signals, you're smoothing noise into your signal and making it more difficult than if you're actually smoothing on the 2-dimensional cortical sheet. Okay. So a while ago, I published something called the Siptify project, which was a tool for trying to put, to try and make all of this a little bit easier, to make a BIDS app that would pre-process data on the cortical surface with much different data types that the current tools weren't working with. And then I needed to learn a lot about a lot of different software things back in the day. But so I said that those points were all useful, especially. I'm gonna go back. I said that smoothing is bad and 2-dimensional cortical registration is better. But is it that much better? Is kind of the question that I still kept getting back from everybody and myself. Is it worth using this extra tool when it's easier, I already know how to do kind of a three-dimensional approach to brain science. So my last shout out is a paper that I did looking at schizophrenia case control differences and just comparing different cortical surface, cortical signal extraction methods to see what the difference would be. So I basically pre-processed the data the exact same way, and I just extracted from the cortical surface differently. So I used a volume-based tool, very similar to what some other papers would do, where I just took spheres in different areas of the brain. I took a surface-based approach, so I took the surface pre-processed data, and I took circles, so the equivalent, across the brain, from different areas across the brain. And then I had a third approach, which was more similar to something like hyperalignment, where I tried to use the functional data to further change that. I'm not going to talk so much about the third, but this just shows individual level correlation matrices from those three different techniques. You can kind of already see at the individual level that you're getting a different correlation matrix just by the cortical extraction tool. And these are those correlation matrices at the population mean level. You can already start to see, and this is statistically significant, there's a little bit more network structure actually using the surface-based approach than the volume-based approach. And actually there's a lot more network structure with the personalized approach, but that's what the personalized algorithm is optimized for, so that makes sense. What was more interesting to me was to think about the differences between schizophrenia and healthy controls and whether or not those results would change with the cortical sampling technique and to what extent. So, okay, I have a very fancy graph that I was very proud of a few years ago, where I decided to take the correlation matrix and plot it in a different way, or differences. Okay, so along the middle is all your regions of the brain. All your cortical regions are up here. These three regions are three subcortical structures that are really important: the striatum, thalamus, and cerebellum. Every time you see a blue arc, so every blue arc in this picture is an area where there's hypoconnectivity or less connectivity between the more connectivity in the health controls and the schizophrenia. The red arc means there's more connectivity in the schizophrenia group than in the health control group. Now, this is with the volume-based template, so this is the circles. This is with the surface-based approach. And this is with the personalized approach. So you can see that the arcs are moving, they're changing a little bit. If you put it all together, there's actually more arcs, so more significant edges in a surface-based approach than the volume-based approach, and there's even more in a surface personalized approach. You can also see a little bit more interpretable network structure as you go from left to right as well. Thanks to a reviewer, they asked me to try a different way, so I actually compared the shaper volume approach, the shaper surface approach, and I got kind of an even stronger result where you actually have twice as many significant edges when you move to the surface approach than when you use the volume approach from the exact same input data. Okay, so but to end off, surface-based approaches are great. They are important. You can use my tool, but now actually the field has caught up with me. Our amazing developers in the field have caught up with me, so you can actually just use the proper flags in fMRI prep to get similar results to what I have, as well as there's a new XCVD tool, which does amazing, better post-processing than I was doing, so I'm switching to those two. Surface-based approaches are great for pretty pictures, but also because their volume is better than surface, and you can get stronger predictions in clinical models. Okay, that brings together the whole thing. So today I told you about parsing heterogeneity that was important to people with disorders, looking upstream, so thinking about how we can predict the future, or looking longitudinally across people and using the tools of reproducible science. One lasting I just wanted to last on things we need in the future is actually, I've been thinking a lot more about longitudinal methods and looking upstream. And one huge problem is that I'm searching for longitudinal analytic tools, and it's hard. So it feels like all the cool computational techniques are meant for cross-sectional data at this point. So if anybody out there wants to do some more development in the longitudinal space, that would be awesome. And with that, thank you for listening to me talk, and thank you to the amazing people in my lab, the amazing fighters.
Thanks, Erin. Thanks a lot. Any question for Erin? Or maybe online?
I'm far from the microphone.
Oh.
Can you just say that if somebody online wishes
Yeah.
to ask a question, that they can turn on their microphone or put it in the chat?
Yeah.
Thank you. If you have a question, yeah, maybe repeat that so that the microphone is.
Celia, we heard that.
Great.
I had a question about the TASTE study.
Yeah.
So you mentioned that 48% of your cohort, function controlled through the PSS.
Mhm.
Have you looked at what the, like the percentage of, like a control study would be for a PSS, like threshold? Like, do you have those numbers for a comparative?
It's hard to say, so that number came from Philadelphia neurodevelopmental cohort, so that idea of the 1, 6 or 3 5s model, and I think that one was closer to like 10 or 20%.
Okay.
In the community sample that they had, but I think it'd be important actually to start looking at things like, ABCD doesn't have the exact same skill, but it has something similar. To start looking at the general population as well.
I have like a question in the comment. Can you show up the RTMS pipeline?
Yeah.
The one that was supposed to run in less than a week and then inform.
Yeah, that one. This is like an older picture of it too.
Well, okay, while you find it, I was just gonna comment that
Yeah.
essentially you guys are basically doing, ad hoc treatment planning on the fly.
Yeah.
Using EM modeling as well as neuroimage processing.
Mm,
So, I mean, this is like not for RTMS, but for instance, radiation therapy, quite a robustly developed thing, and
mm,
with, your colleagues in the medical physics department, perhaps you could gain insight on how they do these kind of things, because that's all like
Mm.
really full of kind of like a
Mm.
gamma knife or external beam and stuff like that.
mm,
And perhaps even use some of the quite well-developed tools there,
yeah,
import these like, anatomical scans and treatment plans of C clips, and then instead of running, an electron transport, radiation transport algorithm, run this
Yeah.
electromagnetic modeling algorithm there. And that would
Yeah.
be very, very similar to what's already very well established in radiation therapy.
it's true. I don't know if.
I'm a physicist at the
Yeah.
at Toronto Western. It's never
Yeah.
all the people
Yeah.
that study broken PMH.
It's true.
I
That is
just-
actually, yeah.
Yeah, sure.
Mhm,
It was somewhere near the beginning, you
Yeah.
talked about partial least squares,
Yeah.
which is obviously a linear method.
Mhm,
What do you think is the scope for non-linear methods?
it's true. That's what. Yeah. I totally want to look a little bit more at the nonlinear methods, because things like diffusion map embedding. The diffusion map embedding, which is a more nonlinear method, did give a very different result to that, give us a different understanding of the brain relationship matrix than some of the linear decompositions that have happened before. I am actually, maybe we'll have more understanding, because I think Richie does want to compare kind of her PLS methods, or even PLS at a larger level, to some of these nonlinear solutions. But I do think nonlinear solutions will help.
I guess it depends a little bit on the number of subjects that you have and those things, because, I think you had something like 1,000 subjects, you have,
Even less, yeah.
even less, so
Mhm.
we start to get, a bit, probably,
Yeah,
the chance of overfitting those things,
Yeah.
is very high.
Yeah,
Given the heterogeneity, I guess, so getting some linear trends is probably a good. And there's, like a lot of, there's been a couple of papers and, Michelle, so
Yeah.
working on that as well, looking at the stability of those things,
Mhm.
with respect to sampling of the,
yeah,
so I think it's, you might want to
Mhm.
connect to that as well.
yeah. I think there's also something about the strength of what you're decomposing.
It depends on how much you know.
Yeah, how much signal is there to decompose. Yeah.
And kind of a stats question,
Yeah.
I guess. When you're doing an analysis on like the data set that you're still collecting, and then I assume some of these things you're gonna repeat,
Mhm.
how do you like? Is there a need to correct for multiple comparisons because you've like used part of the data set already,
Mhm.
and then you're using more of it?
Like, in terms of that, personalized method, you're saying, or?
And I think that for the TASE study,
Yeah.
you've done some.
Yeah, yeah, I think right now, especially, those are all kind of preliminary. I
Yeah.
don't remember how much correction, comparison there was, but we're more interested in preliminary effect sizes, and how much those things relate. And even just the descriptives at this point, because it was actually really interesting. We tried to find like what was the expected level or what was the basic level, and we realized that like, because when you see your clinician, they have like 10 minutes with you, they're really only looking for presenting symptoms. We actually didn't have a baseline understanding about how many multiple diagnoses were coming in, and simple things like that.
Okay, not quite what I meant.
Yeah.
I can chat with you.
Yeah.
There's a question in the chat, from Suresh. Talking about brain age.
Mhm. Yeah, okay. Yeah, so actually that is,
So maybe repeat the question.
so a very naive and general question. Brain age is a great metaphor, but is it also a good unidimensional way to capture a range of changes in MRIs? And actually that's my main reason for wanting to do it, which is more of a data production technique. So, yeah, the thing that I really love about those models is that they are multivariate. But they give you one number, and one number is very useful when you have a lot of other dimensions that you might want to be looking at phenotypically, to look at. So having some kind of large constructs like brain age, I think are really great ways to start doing our analysis.
But then it's never clear to me if I can
Yeah.
follow up on that. So brain age, you take into account sex, or then, should you take into account like, the next variable that is the most impactful,
Yeah.
like, how, at what stage do you stop, like, subtyping or
Yeah.
doing,
Yeah.
so there's a continuum there, or at least that is a bit unclear to me. The theoretical framework is a bit unclear in that respect to me. I don't know if you have thought on that.
Yeah, I think we're getting close, even if you go beyond sex to gender identity. The next thing over, which is becoming a larger issue to really understand from the TASE sample, because there's so many.
But like maybe brain age
Yeah.
curve would be fairly different to, like, with the high SSE versus
Yeah.
low SSE, or maybe like
Yeah.
those variables and the number of variables that you really need to collect
To collect.
to have, like, a fair understanding, and
Mhm.
possibly the number of redundant variables.
Yeah.
Because it's a bit daunting to me in
Yeah.
some ways. Like, we see a lot of studies with maybe many variables, but like 1,000
Yeah,
subjects, and it's never clear how
yeah,
that's
which
going
one,
to be
which ones to bring in?
how's
which
that
one
going
is-
to be
Yeah,
replicated.
Well, I know the students have time with you, during lunch,
Mhm.
so maybe this is a good moment to, thank you so much for one
Yeah,
presentation.
it's very inspiring.